Hello learners, today we are going to learn immune system. The animals and the human beings are forced to encounter many dangerous microbes in their daily basis life through water, through air or through food. Inside our body there is an amazing protective mechanism which is called immune system. It works silently in our body. The immune system is designed to protect us from millions of microbes, toxins, parasites. The immune system protects us in three different ways. One, it creates a barrier that prevents bacteria and viruses from entering the body. If a bacteria or virus does get into the body, the immune system tries to detect them and eliminate it before make itself dangerous. If the viruses or bacteria are able to reproduce and start causing problems, our immune system is in charge of eliminating them. There are other components of the immune system which include spleen, lymph system, bone marrow, white BC, antibodies, hormones and complement system. There are two types of mechanisms in immune system. One is non-specific defense mechanism and other is specific defense mechanism. Other classifications is first line of defense mechanism and second line of defense mechanism. First line of defense mechanism includes skin's layer, mucosal membrane, secretions of skin and mucosal membranes, skin and gut bacteria. Second line defense mechanism includes phagocyte, inflammatory reaction, fever, natural killing cells, complement systems and interferes. Those all mechanisms we have discussed all are non-specific. Specific mechanism includes antibodies and lymphocytes. Non-specific defense mechanism has one part which is called external defense mechanism. The external defense mechanism comprises of physical and chemical barriers to entry of pathogens. Physical barriers, skin, mucosal membrane, and chemical barriers is skin secretions, saliva, gut secretions, bile, tears, nasal secretions, vaginal bacteria, etc. The internal defense mechanism done by RBCs and white BCs. What is macrophages? They are large, long-living, effective phagocytes which enlarge out of monocytes. It is an irregular cell about 25 to 50 mm in size. It has a large ovoid nucleus extended on this site. Macrophages are of two types, fixed types and wandering types. Fixed types are located permanently in certain organs like lungs, liver, renal glomeruli, brain and connective tissues. Functions of macrophages. Phagocytosis. Phagocyte the microorganisms and their inner particles. Secrete IL-1, interleukin 1, TNF, tumor necrosis factor, GCSF, granulocyte colony stimulating factor, and MCSF, macrophage colony stimulating factors. Process and present the antigen to immunocompetent cells, destroy old RBC, and initiates catabolism of hemoglobin. The next internal defense mechanism point is inflammatory responses. Injury, cut, or bite of an animal brings foreign material like bacteria, viruses, fungi, etc. to enter the tissue. Certain substances released by the damaged cell. For the initial formation of the blood clot, clotting checks the flow of the blood. Next is pus formation. Once a neutrophil finds a foreign particle or a bacteria, it will engulf it by releasing enzymes, hydrogen peroxide and other chemicals from its granules to kill the bacteria. In a site of serious infection where lots of bacteria have reproduced in that particular area, pus will form. Pus is simply dead neutrophils and other cellular debris. Many neutrophils and macrophages are killed by the toxins released by high virulent species. Like pus formation, the next internal defense mechanism is fever. We have seen that due to injury, cut, bite, or inflammatory responses developed, and this is located in a systematic way. The localized response is confined to the site of injury only. The systemic response affects the body in the case of severe infection or severe injury. In that case, the white BC count increases, body temperature rises, causing fever. Next is complement system. This is a series of proteins. There are only a handful of proteins in the complementary system, and they are floating freely in our blood. The complementary protein are activated by and work with the antibodies. Hence the name. They causes lysing of the cell and signaling to the phagocytes that a cell needs to be removed. Next, interference. It acts with the viruses like antibodies, complementary proteins, and their job is to lead signals to the one cell to another cells. When a cell detects interference from other cells, it produces proteins that help prevent the viral replication in the cells. Next topic, specific defense mechanism. First, measure histocompatibility complex, which is called MHC. MHC molecules are important components of the immune response. They allow cells that have been invaded by the infectious organisms of the immune response, which is detected by the cells called T cells, T lymphocytes. The MHC molecules do this by the presence of fragments of proteins called peptides belonging to the invader on the surface of the cells. The T cell recognizes the foreign peptide attached to the MHC. Therefore, MHC is a set of genes that code for cell surface glycoproteins. Glycoproteins mark the body cells as self and are divided into two main cases. One is MHC1 molecules are found on the surface of almost every cell of the body, and two MHC molecules are confined to the few specialized cell types. Next is antibodies. Antibodies are produced by the plasma cells. They are Y-shaped protein that each responds to a specific antigen. And now we have to know about antigen. What is an antigen? The foreign matter like bacteria, virus, or toxin that enters to the body and elicits the specific immune response by lymphocytes, that is called antigen or immunogen. The molecule of antibody has two pairs of chains like structure and it is linked by the disulfide bond. The two longer chains are called heavy chains and two shorter chains are called lighter chains. Two long and two short chains are identical. The antigen binding site is at the N-terminal position, which is the end of the polypeptide chain. The site of complementary fixation is towards C-terminal portion. There are the differentiation between antigens and antibodies. Antigens is the foreign material, elicits antibody formation and antibody synthesis by the organism to combat that foreign material called antigen. It is a protein and polysaccharide molecule, antigens, and antibodies is the immunoglobulin. Antigen occur on the surface of the microbes or as a free molecule, and antibodies occurs on the surface of the plasma cell and also in the body fluid. Antigen binds to the macrophages to reach a helper T cell to initiate immune response, and antibodies directly join to the antigen to destroy it later. Antibodies classified in the 5 classes. Those are immunoglobulin A, immunoglobulin D, immunoglobulin E, immunoglobulin G, and immunoglobulin M. IgA, IgD, IgE, IgG, and IgM. Next is innate immunity. It is referred to a basic resistance to the disease, that is systemic processes, first line of defense against infection. The characteristics of the innate immune mechanism includes responses are broad spectrum, there are no memory and long lasting protection immunity, there are limited recognizing molecules, the responses are phylogenetically ancient. Next topic is phagocytosis. Phagocytosis involves in the ingestion of particular materials including whole pathogenic mechanisms. The plasma membrane expands around the particulate material to form large vesicles called phagosomes. It is carried out the neutrophils or macrophages. Neutrophils are highly effective against pus formation bacteria. We already know that macrophages are tissue phagocytes which are derived from the circulating monocytes. Phagocytosis mechanism. There are two types of mechanism. One is O2 dependent and other is O2 independent. Humoral mechanisms. Besides all the mechanism, there are some humoral mechanism which includes acute phase proteins, interferons, natural killer cells, and contribution of eosinophils. Specific acute immunity or adapted immunity. It involves antigens presenting cells, activation and proliferation of the antigen specific B lymphocytes, activation and proliferation of the antigen specific T lymphocytes, and production of the antibody molecules, cytotoxic T lymphocytes, and the cytokines. The human body can respond to antigens in many different ways. There are two major categories. One is antibody mediated immune systems, AMIS, by circulating antibodies, also known as humoral immunity. Cell mediated immunity system, it is sensitive system known as cell mediated immunity. We all know the primary lymphoid organs are thymus and bone marrow. Secondary organs are lymph nodes, spleen, and lymphoid tissue. When an AG microorganisms enter in the body, one of the secondary lymphocyte organ traps it and mounts an immune response. An exact organ involved depends on the sites of injury. One is tissues, bloodstream, and mucosal surface. Antibody mediated immune system. What is the specific role of antibody mediated immune system? It defines the body against free viruses, bacteria, and polysaccharide capsules and toxins that enter in the body fluid or the limb. Antibody works as different ways. One is neutralization, two is agglutination, three is precipitation, and four is opsonization, and last is complement activation. Cell mediated immune system is also works against bacteria and viruses which have already entered in the host cells, also against protozoa, fungi, and parasites. It is protects our body by activating antigen specific T lymphocytes, activating macrophages and NK cells, stimulating cells and secreted the cytokinins, etc. Classifications of T cells: killer T cells, helper T cells, suppressor T cells, and memory T cells. Role of macrophages in the immune system. Macrophages respond to an infection. They engulf the microbes and then antigen display the antigen to alert the lymphocytes. The antigen is held onto the surface of the macrophage by the protein which marks as macrophages as self. With an antigen flagged on it, macrophage moves to the nearest lymph nodes. A helper T cell recognizes the macrophage's antigen. Simultaneously, macrophages secreted endogenous pyrogens like activates T and B cells to grow and proliferate, causes fever and microbial action slowdowns. The leukocytes and development and its regulation. Development of granulocytes. Following stages are there: meioblast, promyelocyte, myelocytes, metamyelocytes, band cells, and mature granulocytes. Growth factors which affected granulocytosis: it's interleukin 1, tumor cell factor, granulocyte macrophage colony stimulating factor, interleukin 3 on the site of injury. Sites of injury: one is tissues, bloodstream, and mucosal surface. Antibody mediated immune system. What is the specific role of antibody mediated immune system? It defines the body against free viruses, bacteria, and polysaccharide capsules and toxins that enter in the body fluid or the limb. Antibody works as different ways. One is neutralization, two is agglutination, three is precipitation, and four is opsonization, and last is complement activation. Cell mediated immune system is also works against bacteria and viruses which have already entered in the host cells, also against protozoa, fungi, and parasites. It is protects our body by activating antigen-specific T lymphocytes, activating macrophages and NK cells, stimulating cells and secreted the cytokinins, etc. Classifications of T cells: killer T cells, helper T cells, suppressor T cells, and memory T cells. Role of macrophages in the immune system. Macrophages respond to an infection. They engulf the microbes and then antigen display the antigen to alert the lymphocytes. The antigen is held onto the surface of the macrophage by the protein which marks as macrophages as self. With an antigen flagged on it, macrophage moves to the nearest lymph nodes. A helper T cell recognizes the macrophage's antigen. Simultaneously, macrophages secreted endogenous pyrogens like activates T and B cells to grow and proliferate, causes fever and microbial action slowdowns. The leukocytes and development and its regulation. Development of granulocytes. Following stages are there: meioblast, promyelocyte, myelocytes, metamyelocytes, band cells, and mature granulocytes. Growth factors which affected granulocytosis: AIDS, interleukin 1, tumor cells factor, granulocyte macrophage colony stimulating factor, interleukin 3, gamelocyte colony stimulating factor, macrophages and inhibitor factors.